All right. Good afternoon, everybody. Welcome to one of our final research conferences for the summer. I know we are all very anxious for summer. We have a few more of our Mondays left together. And I'm really thrilled today to present Tom Lagace. So Tom has been here at the Institute since approximately 2009 or so. We hate when we have those dates announced for everybody, so we can, we're just been a couple of years, right, Tom? Um, he's an associate professor in the Department of Biochemistry at the University of Ottawa. He got his PhD in molecular biology and biochemistry at Dalhousie University, where he studied phospholipid metabolism. And so Tom has been studying lipids for a very long time. He then went on to the Department of Molecular Genetics at the University of Texas Southwestern Medical Center, or UTSW, in Dallas. And that time where Tom joined UTSW was actually one of the most pivotal times, probably looking back in history, of what the understanding of lipids and lipoprotein metabolism, and one of the critical times to get to where we are now in our ability to manage hyperlipidemia. And that's because at that time, they had just discovered this kind of interesting protein that was a real mouthful to pronounce, PCSK9. And if you haven't memorized the order of the letters, the P and the S and the K and the 9, you fumble all over it, but Tom has memorized it because he has been studying it ever since. And at that time, it was one of the, amazing examples of how a combination of human genetics and molecular biology and biochemistry really led to the discovery of one of the most important medicines we've had in the last decade. And that is drugs that inhibit PCSK9. They're widely used. We prescribe them in this institute every single day. But those would not be possible without the type of work that Tom did at that time at UT Southwestern, as well as ever since in his own laboratory. And the story doesn't stop there. Just because we have medicines to treat, in this case, PCSK9, doesn't mean we understand. Most of the time, actually, it's quite the opposite. Often we end up having a medicine first before we fully understand how something works. So this is what Tom's lab has been doing for the past 15 years or so, since he's been here at the Institute, is understanding bit by bit how PCSK9 works. And shockingly, as he'll tell us today, it's far more complex than ever thought before. It is not just a regulator of lipids and lipoproteins. It does much, much more. So we are excited to see this work that, of course, is not only pivotal, as I mentioned, in the, in understanding clinically, but I know we're going to see some really cool things happen from the understanding that Tom and his lab have are contributing. So welcome, Tom. Welcome, Dr. Lagace, to the podium.
Thank you, Katie. Can everyone hear me okay? Get right into it. Okay, so I'm going to be talking today about PCSK9, well, my favorite protein, as Katie mentioned. I've been studying it for a long, long time. And we're especially excited about new collaborative efforts here at the Heart Institute looking at the evolving role of PCSK9 in inflammation, which has come out, Katie really hit the nail on the head. Sometimes we come up with a medicine first, and then from that medicine, we learn a lot about the function of the protein. So that's exactly the story that I'm going to be telling you today. Okay, so here's an outline of what I'll be talking about. Usually I spend the entire talk talking about PCSK9-mediated LDL receptor degradation, but this is going to be a small part of the beginning of the talk, and then talk about some of the strategies that are currently out there for cholesterol-lowering therapies that target PCSK9. And then I really wanted to spend most of the talk talking about LDL receptor independent role of PCSK9 in inflammation, which has come out of these clinical studies, and specifically a potential role of furin proteolysis of PCSK9 in these processes, and also the role of PCSK9 LDL binding, which is something we study in our laboratory. We've been doing quite a bit of work in the last number of years on the molecular basis of this PCSK9 LDL interaction. because we believe this could be a novel target for inhibiting PCSK9, not only in its ability to mediate LDL receptor degradation, but also in these pro-inflammatory processes that it's involved in. All right, so there's a pivotal role. I don't need to tell everyone in this audience about the pivotal role of LDL cholesterol in atherosclerotic lesion development, but it begins with the transcytosis of LDL particles across the endothelial layer from the bloodstream into the subendothelial space. The LDL particles can be modified through processes such as oxidation and aggregation, and these modified LDL particles get taken up by macrophages. Unlike other cells that would downregulate receptors and modulate their ability to uptake LDL to maintain cholesterol homeostasis, these scavenger receptors are not downregulated, so they keep taking up the LDL cholesterol to eventually the point where they become engorged in cholesterol and what makes this sort of foam cell. These can undergo types of cell death, necrosis, necroptosis, that can release pro-inflammatory factors that will recruit further monocyte infiltration in the subendothelial space, sort of a perpetuating cycle in the growth and development of the atherosclerotic lesion in the subendothelial space. Now, this is a process that occurs in all individuals and under these types of mechanisms, but the rate at which it occurs and the stability of the plaque is a function of an individual's risk factors. Now, one of these risk factors is the concentration of your circulating LDL cholesterol. Obviously, if there's more circulating LDL cholesterol, there's more of it that's going to make it into this space and drive this progression of the atherosclerotic plaque. So there's lots of development of cholesterol-lowering therapies. Probably all know about the statin drugs that raise the levels of the LDL receptor. Ezetimibe is a drug that will prevent or inhibit the absorption of cholesterol through the diet. And then the newest player are these PCSK9 inhibitors that inhibit PCSK9 to raise LDL receptor levels. So not only does LDL have this sort of accumulation effect in macrophages, but it also has potent signaling effects on pro-inflammatory processes. So oxidized LDL is something known as a danger-associated molecular pattern. So this can bind to these scavenger receptors that not only take up that cholesterol, but they also signal through various mechanisms that will lead to pro-inflammatory processes, that some of which I'm listing here in this diagram. So oxidized LDL can be captured by these scavenger receptors such as CD36, another one called lectin-like oxidized LDL receptor 1 or LOX1, and this induces this pro-inflammatory signaling. And it's sort of a perpetuating domino effect. As you get more and more of this happening, there's more oxidized LDL being formed in the subendothelial space, more pro-inflammatory factors being released, and more recruitment of monocytes, and on and on and on. So this is sort of a perpetuating cycle that's occurring within the atherosclerotic plaque. So a lot of what we know about LDL cholesterol levels in the blood plasma and its relationship to atherosclerotic heart disease is through studies of single gene disorders. So these are genes that are involved in the uptake of the cholesterol-rich LDL particle in the liver, one of which is the LDL receptor that binds to the apolipoprotein B100 moiety of both IDL and LDL. And in individuals that have the highest level of LDL cholesterol in an inherited fashion, this is a disease known as autosomal dominant hypercholesterolemia. You also hear it referred to as familial hypercholesterolemia. So this mainly occurs through gene mutations in the LDL receptor itself, or its ligand, ApoB100, which is on the LDL particles. And then in 2003, a new gene was discovered. So these were in families that had autosomal dominant hypercholesterolemia, but they didn't have gene mutations in the LDL receptor or ApoB100 genes. And a new gene had been discovered by a Canadian, Nabil Seda, called PCSK9. And it was found in a location which this group in France had identified as a novel locus for this disease, autosomal dominant hypercholesterolemia. They sequenced that locus and found genetic mutations in the PCSK9 gene that were found to be causative for autosomal dominant hypercholesterolemia. So this occurred in 2003 in this landmark study. All right, so Since that time, many gene defects gene mutations have been identified in the PCSK9 gene. These either cause a gain of function. So those were the types that were found in these families in France. So they result in a gain of function of PCSK9, causing an elevation of LDL cholesterol in the blood plasma and an increase in your cardiovascular heart disease risk. There's also loss of function mutations. These people are quite lucky. They have lowered LDL cholesterol and a lowered heart disease risk. I'm just listing a few of these. There are actually numerous mutations of both types found throughout the PCSK9 protein. And one of the things that our lab does is we try to understand the mechanisms behind these mutations, because that gives us good functional insights on how PCSK9 is working. Right, so in the early days, we were able to establish that PCSK9 causes LDL receptor degradation through this pathway where it gets secreted out into the extracellular space, at which point it can bind to the LDL receptor. And unlike the LDL particle, which releases from the LDL receptor in the acidic environment of the early endosome, which allows the LDL receptor to recycle back to the cell surface, PCSK9 binding is actually tighter at acidic pH. So it doesn't release, it doesn't let go. And it's still unknown how this occurs, but after this case, both the PCSK9 and the LDL receptor are directed to the lysosomes and they're degraded. And so there's less LDL receptors on the cell surface. Now, as a secreted factor, PCSK9 might also be regulated by factors within the circulation. So that's something that our lab is especially interested in, because these physiological regulations might give up new novel ways in which to inhibit PCSK9. One of which we've found is the binding of PCSK9 to LDL particles themselves. Okay, but initially from these studies of this pathway, it became clear one way in which you could inhibit PCSK9, that is through the development of blocking antibodies that would prevent the binding of PCSK9 to the LDL receptor. But first we had to identify what that interaction site was. And so we were able to show that the region of LDL receptor is a part called the epidermal growth factor-like repeat A, or EGFA domain of the LDL receptor. And you can produce these little fragments of the LDL receptor alone in bacteria. So what we did was produce this, and then we're able to get a co-crystal of PCSK9 bound to it. And this way, we're able to show that the catalytic domain of PCSK9 was the part that was binding to the LDL receptor. And one of the interesting things we found in this study, quite by accident, is there's this intrinsically disordered region in the N-terminus of the PCSK9 prodomain. It was getting in the way in our co-crystal formation, so we just took it off. And then we were surprised to find that this particular form of PCSK9 lacking this N-terminus was much more active at binding to the LDL receptor, bound with about sevenfold higher affinity. So this also identified this region as an autoinhibitory domain of the LDL receptor binding, because it's located far away from the binding interfaces you can see here. So this is also something that we've been studying since then. So with this strategy, then the drug companies all raced to be the first ones that could develop these monoclonal antibodies that would target the catalytic domain of PCSK9. And they had these binding assays that were developed where you could use the EGFA domain of the LDL receptor in a plate assay to screen all these antibodies. And so the first ones that were developed were by Amgen and Regeneron, and these have been approved since 2016 for use in patients. So as Katie mentioned, this is very rapid development of a pharmaceutical therapy that originated from human genetic studies. So it's a real success story of this sort of bench to bedside approach. And so some of the highlights of these use of these antibodies, up to 70% lowering of LDL cholesterol in hypercholesterolemic patients. So this is very, very potent at lowering LDL cholesterol, much better than, say, the statin drugs can do or other treatments. There's also significant reductions in major adverse coronary events, even in patients that have existing coronary artery disease. So these are very effective, not only in LDL cholesterol lowering, but also in lowering cardiovascular events. The problem is that there's a very high cost to these biologics, about $15,000 per year to treat a patient with hypercholesterolemia. So this really limits the number of patients that can get access to these potentially life-saving therapies. So the pharmaceutical industry is very interested, of course, in finding alternative ways to inhibit PCSK9, one of which has been approved since 2020 is an siRNA. So this is, again, an injectable type of a treatment, but now you only have to use this twice a year instead of twice a month, like the monoclonal antibodies. So it's less expensive. And this targets the liver expression of PCSK9. So PCSK9 is expressed throughout the body, but the major source of circulating PCSK9 is the liver, right? There's also a couple of small molecule inhibitors that are now in clinical trials. One is a macropeptide, which blocks the binding interface of PCSK9 to the LDL receptor. This took about 15 years to develop from the time of that co-crystal study and just shows the real big challenges here because the binding interface is very flat and doesn't have any obvious druggable pockets for these types of small molecule blockers. There's also this sort of mysterious one that's in clinical trials. I haven't found any information on what this does, but they claim that it binds to a pocket in PCSK9 and then inhibits LDL receptor degradation. This is now in phase 2 clinical trial being developed by AstraZeneca. Another approach, which I find a bit alarming, is that they're using CRISPR gene editing tools to basically permanently knock out PCSK9 gene within the liver. And of course, this has raised some concern about the possible safety effects of targeting a protein that you don't totally understand its physiological role. And so especially given that there's millions of years of human evolution, clearly if this protein wasn't needed, we would have gotten rid of it by now. So one of the reasons in which there's this alarm is that through the clinical trials of the PCSK9 monoclonal antibodies has emerged this evolving role of PCSK9 within innate immunity. All right, and this comes from studies in which the PCSK9 monoclonal antibodies have been shown to be effective in all kinds of various conditions related to the immune system, such as cancer, sepsis, and various inflammatory conditions, such as autoimmune diseases. And up to this point, these role of PCSK9 within these inflammation and immunity processes is not very well understood. So there's several theories that are out there about how it might be doing this. One is that PCSK9 could be a general degradative chaperone. So there's evidence that PCSK9 can bind to other cell surface proteins and other receptors in addition to the LDL receptor. Some of these are very closely related. These are part of the LDL receptor gene family. So they also have epidermal growth factor-like domains. And there's some evidence that PCSK9 binds to them much worse than it binds to the LDL receptor, but there's still some binding. A lot of these other ones, though, are not related, all right? And for instance, these scavenger receptors, CD36, LOX1, have no structural relationship to the LDL receptor. So while PCSK9 could bind to these, and then there's this theory that it sort of links to a general degradative machinery through a putative protein X in the endosomal pathway, and that anything PCSK9 is bound to will be directed to the lysosomes for degradation. However, there's no evidence so far that any of these interactions would be blocked by PCSK9 monoclonal antibodies in the same manner that PCSK9 blocks the LDL receptor interaction. And then a second theory is that PCSK9 itself is a pro-inflammatory signaling molecule. So PCSK9, this is sort of a provocative editorial that accompanied this really nice article that was published in Circulation in 2021, suggesting that PCSK9 itself could be one of these danger-associated molecular patterns and bind to these signaling receptors to induce pro-inflammatory pathways. And this came from a study in which they found that PCSK9 was involved in platelet activation, thrombosis, myocardial infarct expansion through binding to CD36 on platelets. And the way they did this is that in LDL receptor knockout mice, there's an elevation of plasma PCSK9. And that's because the LDL receptor is the main conduit through which PCSK9 gets cleared. It binds to the LDL receptor and gets taken out of the plasma. So in LDL receptor knockout mice, PCSK9 levels build up. So they found that in the LDL receptor knockout mice compared to wild type mice, if they induce platelet aggregation with this ferric chloride treatment, this could be prevented by the PCSK9 inhibitory monoclonal antibody evolocumab. Right, so this is very important to show that there was this pro-inflammatory effect that was independent of the LDL receptor. All right, they also interestingly showed that PCSK9 could be found in thrombi isolated from human patients that had undergone myocardial surgery for myocardial infarction. Right, so these methods that they detect PCSK9 are ELISAs, immunohistochemistry, that don't really detect different forms of PCSK9 that are found in the plasma. And we found that in LDL receptor knockout mice, most of the PCSK9 is found in a cleaved form, a truncated form that results from furin proteolysis. In fact, almost 60% on a normal chow diet and over 85% of it in a high-fat diet is in this furin cleaved form, right? So that raises the question, is the furin proteolysis form of PCSK9 having some type of a function in inflammation? So a little bit about this at first. So it cleaves PCSK9 at a region in the catalytic domain that overlaps the LDL receptor binding site. All right, so furin cleaved PCSK9 can't bind to the LDL receptor. And for this reason, maybe it's been overlooked because people think of it as just being inert. Right, it doesn't bind the LDL receptor. It probably just doesn't do anything. So about 10 to 40% of PCSK9 in normal human plasma is in this furin cleaved form. And but interesting, a study had found, and maybe not surprisingly, that truncated form of PCSK9 went down in patients that were treated with the PCSK9 inhibitory monoclonal antibody evolocumab. But again, this is sort of just an aside in this paper. Oh, by the way, this happens, but no, not really thinking about what it could be doing. So we've also seen that another antibody, aliricumab, blocks the cleavage of PCSK9 by furin, which you can see in this sort of in vitro assay. So these antibodies are very effective at inhibiting the proteolysis of PCSK9 by furin. So does that have any pro-inflammatory role? So first off, with help from Aaron and Natasha, we got some plasma samples from mice that were either male and female mice that were treated with saline or with lipopolysaccharide for 24 hours to reduce an inflammatory condition. And others had shown in the past that LPS or other inflammatory stimuli raise the levels of plasma PCSK9 through increased mRNA expression in the liver. But again, they hadn't really looked at the different forms of PCSK9 that are found within the plasma. So we saw that, yes, as expected, PCSK9 levels went up in the plasma in mice treated with LPS. But the main way in which the biggest increase was in the furin proteolysis form. And the biggest increase we saw as well was in female mice, right? So whereas the increase in the uncleaved form of PCSK9 was much less pronounced. So this raises a question about whether PCSK9 role in inflammation and furin proteolysis could explain some of the interesting clinical findings about PCSK9 in males versus females. So circulating PCSK9 levels have been shown to be higher in women than men. And this might be related to differences in estrogen or in estrogen levels in women, because it's shown that postmenopausal women have higher circulating PCSK9 from premenopausal. And there's an inverse relationship between estrogen levels and PCSK9. And in the clinical trials of the PCSK9 monoclonal antibodies, it's been shown that these are much less effective at LDL cholesterol lowering in women compared to men. But they're very effective still at reductions in these major adverse coronary events. So this perhaps implies that women have a higher propensity towards the pro-inflammatory effect of PCSK9. And since the pro-inflammatory effect is inhibited by these monoclonal antibodies, along with the cholesterol lowering, they have these similar reductions in the major adverse cardiovascular events. All right, so we want to look at whether furin-cleaved PCSK9 is pro-inflammatory. So we're lucky here at the Heart Institute that we have expert collaborators right on site that have a common interest in how PCSK9 causes inflammation. And so we're starting to work with Dr. Rayner and Dr. Ramet, along with Victoria Laurent, who's a postdoctoral fellow in their labs. And luckily, Victoria did her PhD studies on PCSK9 in France, including the LDL receptor independent effects of PCSK9. So perfectly in line with what we want to do here. So this is a plan we have. We're going to do some in vivo studies where we're going to overexpress either the wild type PCSK9 or one in which there's a furin resistance. So this introduces a mutation in the furin cleavage site, so it's no longer affected by furin. And then in male and female wild type mice, we're going to inject these AAVs. And we're going to do this under a background in which we've also used CRISPR AAV to knock down the LDL receptor gene as well as the endogenous PCSK9. So therefore, we're looking in an environment where the LDL receptor expression in the liver is not there, so that there's not an effect of circulating cholesterol that we have to take into account. So in a short-term study, we're going to look at LPS treatment on parameters of inflammation in these mice. And then in a longer term study, going to look at the effect of high-fat diet on inflammation and atherosclerosis development. And also we're going to do some cell culture studies looking at the effect of either furin-treated wild-type PCSK9 or this resistant mutant on measures of inflammation in primary bone marrow-derived macrophages. So Victoria's been working very hard on this up till now, and I just want to show one of her studies here. She's coming up with data on a weekly basis. That's really great. This is so here she's taken this. So here we can see the input. These are the purified PCSK9. You can see one of them is truncated. This is the RS variant. So this is PCSK9 that has the R218S mutation, or the wild type one. And both have been treated with recombinant furin, and then the furin has been inactivated, right? And so then she adds these to cells overnight. And so this is a process called efferocytosis that's being measured. So here the bone marrow-derived macrophages act in a phagocytic manner to take up apoptotic cells. So these are other cells that have been treated with staurosporin to induce apoptosis. And these dead cells are labeled with a red dye that can be seen through phagocytosis and uptake in the other phagocytic cells. So this is a very important anti-inflammatory process within atherosclerotic plaques for the removal, inert removal of dead cells through this very important process, efferocytosis. So this is a very important anti-inflammatory process that seems to be inhibited by both the uncleaved and the cleaved PCSK9. All right, so next we're going to be looking at important levels of important receptors that are involved in the process of efferocytosis, namely TREM2, MERTK, and LRP1. And these last two are ones that have already been identified as being downregulated by PCSK9, but nobody's yet looked at the effect of furin-cleaved PCSK9. And TREM2 is a novel one that Victoria is very interested in looking at because this is important not only in efferocytosis in atherosclerotic plaques, also it's very important in brain in processes such as Alzheimer's disease and other neurodegenerative disorders. Okay, so we're also looking at the uptake of PCSK9 in either the brain, different types of macrophages. So here we see that in the bone marrow-derived macrophages, there's equal uptake of both the uncleaved and the cleaved forms of PCSK9 in both these unstimulated macrophages and also ones that have been polarized to induce a pro-inflammatory phenotype. So there's really no difference in the uptake in this model, and that kind of fits with the previous data on efferocytosis that both of the forms of PCSK9 are effective. Interestingly, in THP1 macrophages, there seems to be a preference of the uptake of the cleaved PCSK9 over the uncleaved one. And then looking further at this, it's known that some receptors, some scavenger receptors are upregulated in M1 polarized THP1 cells, one of which is this LOX1 scavenger receptor. So I've gotten a plasmid of LOX1 and then testing its direct binding to PCSK9 through studies in HEC293 cells where we overexpress this receptor and then add the different forms of PCSK9. You can clearly see that the cleaved form, the furin cleaved form of PCSK9 is the only one that gets taken up into cells that are expressing LOX1. So certainly we have some evidence that some cell surface proteins and important receptors such as LOX1 preferentially bind to the furin cleaved PCSK9. And another potential pro-inflammatory effect of PCSK9 is through a lipoprotein, a modified lipoprotein called lipoprotein A, or Lp(a). Right, so a little bit about this at first. So this is an LDL-derived particle, which is enriched in oxidized phospholipids. It has pro-inflammatory and pro-thrombotic properties, perhaps because it has this disulfide linkage to another protein called apolipoprotein A. And apolipoprotein A has some similarities to plasminogen, so it might act as a type of a decoy and interfere with blood clotting and these types of mechanisms. It also is very enriched in oxidized phospholipids, so it has signaling effects similar to oxidized LDL, as well as delivering cholesterol to the atherosclerotic plaque. So it's really a bad thing in terms of atherosclerotic development, all these different things that it's doing. So it's been identified as an independent risk factor of cardiovascular disease. And this landmark study came out in New England Journal of Medicine just last year, showing that along with a couple other markers, including LDL, cholesterol, and C-reactive protein, LPA is an independent predictor of 30-year cardiovascular risk in women. Right? So this is a very, very important molecule in the progression of atherosclerosis throughout a person's lifetime. So there's lots of ways that have been explored to lower LPA, but really there are no effective treatments up till now. But they found that the PCSK9 monoclonal antibodies could lower LPA levels by about 20 to 30%. So this is really one of the only effective therapies for lowering LPA. But the way, again, in which PCSK9 is doing this is unknown. And kinetic studies have shown that the effect is mainly on the level of LPA production rather than its clearance. And that's probably because LPA, through the fact that it's very oxidized and also has this linkage to ApoA, it doesn't bind well at all to the LDL receptor. So even though PCSK9 inhibition raises LDL receptor levels, that's not expected to improve the clearance of Lp(a). And also there's evidence showing that Lp(a) lowering in response to PCSK9 monoclonal antibodies even occurs in people that have homozygous familial hypercholesterolemia, meaning they're lacking functional LDL receptors altogether. There's still an effect on LPA lowering. So does this involve the binding of PCSK9 to the LDL particles, which is what we've been studying? And we've shown in 2013, this is work done by Tanya along with a postdoctoral fellow in the lab at the time, Neil Boulder. They were able to show that about 30 to 40% of PCSK9 is bound to LDL particles in normal human lipidemic plasma, right? And we further went on to show with kinetic binding studies, evidence that PCSK9 binds directly to the ApoB100 protein moiety of the LDL particles. So you can see this is satural binding. The KD is at 300 nanomolar, which is around the same binding affinity of PCSK9 to the LDL receptor. And then in competition studies, we're able to show that there's a sort of one-site binding model of PCSK9 to the LDL particle, again, supporting that it's binding to ApoB100, which is the main protein moiety of the LDL particle. So we wanted to find what part of ApoB100 is binding to the LDL, or is binding to PCSK9. So LDL comes from, as a product of the lipolysis of very low density lipoprotein, a very triglyceride-rich lipoprotein that's secreted by the liver. And then this undergoes lipolysis and other remodeling processes within the circulation, becoming intermediate density lipoprotein, or IDL. And then further lipolysis remodeling finally makes the cholesterol-rich LDL. So at that point, the LDL receptor can recognize the ApoB100 because it undergoes conformational changes during this process. So we also want to see, does PCSK9 also recognize conformational changes in ApoB100? So you can see in these gel shift assays that PCSK9 binds very well to LDL and to IDL, but it doesn't bind to VLDL, right? So there's some type of a conformational change that occurs that allows PCSK9 to bind as it goes along this sort of remodeling process. Our colleague at the Heart Institute, Ross Milne, had developed many antibodies against ApoB100 in LDL. The reason why he did this, because he wanted to map the LDL receptor binding site on the ApoB100. And he had found a couple of antibodies that also bound very well to IDL and to LDL, but not to VLDL. And these map to a C-terminal region of the ApoB. So we tested out some of these antibodies, and sure enough, we found one designated 4H11. It's a monoclonal antibody that very efficiently blocks the binding of PCSK9 to the LDL particle. So ApoB100 is one of the largest, most hydrophobic proteins in the entire human proteome, very hard to work with. I was super excited to see this cryo-EM alpha-fold structure of LDL ApoB100. So this really helps us to kind of guide our studies at this point to find what part of ApoB100 is binding. And you can see this is I've shown the 4H11 site in cyan here. This is in a part of the ApoB100 that forms these paired amphipathic helices that come into this sort of trefoil structure. And interestingly, this overlaps the site where the apolipoprotein A binds to ApoB100. So this is in sort of paired amphipathic helices. You can see the cysteine residue that it attaches to kind of sticking right into the crevice of these two helices. Almost can imagine that it's not very accessible under these conditions. So idea is that maybe PCSK9 binding in this area allows that attachment to occur more efficiently, right? So that's sort of the idea that we're going on, and how PCSK9 might be affecting ApoA. But what does this have to do with furin proteolysis? If furin proteolysis is involved, what would that do? So a PhD student in my lab, Ikuosho Asakia, had been studying the binding of PCSK9 and furin cleaved PCSK9 to lipoproteins. He was able to show that the furin proteolysis doesn't really affect the LDL binding. If anything, it makes it a little bit better. But one of the things that's interesting are some differences that are seen. So the furin cleaved PCSK9, you can think of these three fragments now that are produced. All right. So the prodomain is autocatalytically processed and then binds back onto the catalytic domain. So it already has these two segments. And then the furin proteolysis makes it three segments. And under these conditions, the prodomain tends to dissociate. And it's actually really annoying when you're working with furin proteolyzed PCSK9, because often you'll make it, run it out on the gel, and the prodomain is gone. All right. And then that PCSK9 has to be thrown out because it doesn't do anything functionally. It doesn't bind LDL. It doesn't bind to the scavenger receptors that we're testing. It doesn't do anything. So you have to kind of make it carefully. And Ikuosho was really good at this, in which the prodomain stays bound. Because then it does some really interesting thing. It binds really well to LDL. It binds excellent to oxidized LDL compared to the uncleaved one. And interestingly, the ratio of the prodomain to the catalytic domain goes up in the LDL fraction when you use furin cleaved PCSK9. So that implies that maybe the prodomain is dissociated. So that's one difference of furin cleaved PCSK9 binding to LDL compared to the uncleaved one. You might get some instances where the prodomain dissociates, and that could have some changes in terms of the lipid environment or things like that on the ApoB particle. And one of the ways we think that might be is that the furin cleaved PCSK9 also seems to have these membrane active properties. So Ikuosho did these experiments where he incubated either the uncleaved PCSK9 or the cleaved PCSK9 with liposomes, and then isolated them by centrifugation. So the pellet contains the lipid, and the supernatant doesn't have any lipid. And you can see that the uncleaved one stays in the supernatant mainly, whereas the cleaved PCSK9 tends to bind to the lipids. All right, so these types of properties, these types of biochemical properties could be important in terms of that LPA production that I was talking about. The binding of PCSK9 to that site in ApoB100 might have membrane active properties or otherwise make the ApoA attachment site more accessible. So that's what we're starting to pursue. And this sort of fits as well with studies we've done on the binding site in PCSK9 to the LDL particle. So this is studies that were done, again, by Tanya back in 2013, where identified that a region of PCSK9 that was important to LDL binding was this intrinsically disordered region at the N-terminus of the prodomain. So this is missing in all the crystal structures of PCSK9 up to this point. And you can see it's far away from the LDL receptor binding site. And as I mentioned before, this was identified as a negative allosteric site or an autoinhibitory site, because if you remove it, that greatly improves the binding affinity of PCSK9 to the LDL receptor. So Samantha, who is a PhD student in the lab, did some further studies on this region by doing nested deletion analysis. And she found that, so there's both a hydrophobic region, it's a little bit of hydrophobic residues, as well as a lot of acidic residues within this intrinsically disordered region. So she removed either acidic portion or the hydrophobic portion and found that both of them had a negative effect on the ability of PCSK9 to bind to the LDL particles, suggesting some type of a functional overlap. We then did some prediction analysis of structural predictions, secondary structure predictions, and this said that likely this structure would form a helix. And if you put it on a helical wheel, you can see that it separates. There's a hydrophobic face and a hydrophilic face. So it would tend to form a helix in a hydrophobic environment, which allows the ordering of this structure. Right, so hydrophobic environments, such as the surface of a lipoprotein. Then we did some collaborative computational modeling of the helical region in the full-length PCSK9. And this predicted an interaction of the helix with a C-terminal domain PCSK9 that's enriched in cysteine and histidine residues and a lot of positive charge. So that could interact with the negative charges that are in the intrinsically disordered region when it forms this type of a helical conformation. And interestingly, a lot of the gain of function mutations in PCSK9 that are associated with hypercholesterolemia lined up at this interface. Right? So we tested some of these mutations to see if they would affect LDL binding. And all of them had a very derogatory effect or very, very inhibiting effect. And especially this R496W, which is associated with a very severe form of hypercholesterolemia. We found that it almost abolished the binding of PCSK9 to the LDL particles. And then in other studies, we showed that this protein had normal effects on LDL receptor binding and LDL receptor degradation. Then in other studies involving both Samantha and a graduate student at the time, Angela, they found that there was also another region, PCSK9, that was important to LDL binding. And this, interestingly, lines up on the same molecular plane as the site that was identified in the C-terminal domain. So there's other gain of functions in the prodomain that also greatly inhibit the binding of PCSK9 to LDL particles. And if you introduce one of these mutations, this S127R, into the PCSK9, it's no longer inhibited by LDL particles in cell culture. So normally LDL incubation with PCSK9 inhibits its ability to bind to cell surface LDL receptors, but in a protein containing this mutation, it's no longer inhibited. And interestingly, this was one of the first mutations identified in those patients in France. And it's kind of kind of avoided molecular characterization since then. So one of the dramatic things that it does is it prevents PCSK9 from binding to LDL particles. Okay, so to summarize that then, so 30–40% of PCSK9 in human plasma is bound to LDL particles. And there are these gain of function mutations associated with hypercholesterolemia that prevent PCSK9 from binding to the LDL particle. So this could be the reason in which these gain of function mutations cause hypercholesterolemia, because LDL particle binding would inhibit PCSK9. So if they can't bind to LDL, they wouldn't be subject to that inhibition. And these mutations have also helped us by identifying sites in PCSK9 that are critical to LDL binding. So we want to use that information further to try to better understand the molecular basis of PCSK9 LDL binding. But this was all happening in a hydrophobic environment. But where PCSK9 is we want to inhibit PCSK9 is in an aqueous environment, right? That PCSK9 in the blood plasma is the one that you want to inhibit, the free PCSK9 that's not bound to LDL. So we needed to know what this inhibitory region was interacting with under aqueous conditions. So for this, we turned to a a method called hydrogen deuterium exchange mass spectrometry, in which there's rapid exchange of the protein, the hydrogen ions on the peptide backbone with deuterium in the aqueous environment. And then from that exchange, you can map, and then mapping these out on existing crystal structures, you can show regions that undergo dynamic changes due to things like conformational dynamics or protein-protein interaction. Right, so the site that we found that had the highest difference in hydrogen deuterium exchange when we compared wild-type PCSK9 to one in which this region was removed, this region of the prodomain was removed, was a part of the prodomain, a helical region that's very enriched in arginine residues, right? So this could be the site in which the inhibitory prodomain binds onto to cause a conformational change to lower the binding affinity to the LDL receptor. So another interesting thing about this is that this helical region forms a salt bridge to a D129 residue that we've had previously identified as being important for LDL binding. So Alana, a recently graduated graduate student in the lab, purified a mutant form of PCSK9 in which this aspartic acid has been replaced with alanine. And she was able to show that the LDL receptor binding affinity was increased, though not to the same extent as the full deletion of the intrinsically disordered region. And also this is more potent, this D129A mutant is much more potent at causing the degradation of the LDL receptor. So we think we've identified the negative allosteric site in the PCSK9 prodomain and even gotten it down to the single amino acid stage. This important residue, D129A itself, seems to have an autoinhibitory effect on LDL receptor degradation and binding. So we think that the PCSK9 molecule undergoes these dynamic conformational changes when it goes between an aqueous environment and a hydrophobic environment. So you really can't see these in the crystal structures because this region is missing, right? But we think what happened. Okay. Okay. Yeah. All right. All right, so the timing is getting tight, so I'll go quick. All right, so there's a process called electrostatic steering that might be occurring. So in an aqueous environment, it interacts with the positive charges in this region, whereas in a hydrophobic environment, it moves and binds over here in the C-terminal domain. This allows the LDL ApoB to come in and bind to the negative allosteric site. So the idea being that the ApoB100 does basically the same thing as the intrinsically disordered autoinhibitory domain to cause this sort of allosteric inhibition of PCSK9 binding to the LDL receptor. All right, so maybe I'll just skip that and get right to the inflammation part. So PCSK9 LDL binding is not only important to LDL receptor degradation, but also to this inflammation perhaps. So the idea being that PCSK9 can be converted to furin-cleaved PCSK9, where it can bind to these oxidized LDL particles, and then through this act as a ligand to these scavenger receptors such as CD36, or bind directly by itself to CD36 or LOX1 and other scavenger receptors, and then induce these pro-inflammatory effects within the cells. So the monoclonal antibodies would block this process by inhibiting the proteolysis of PCSK9 by furin. And there's lots of evidence, as I mentioned, clinical studies of the protective effect of these PCSK9 monoclonal antibodies in acute inflammatory conditions such as sepsis, myocardial infarction. There's even clinical trials on COVID-19 with PCSK9 monoclonal antibodies. So why do we want to target LDL binding site in PCSK9? You can think of several important things that might come out of this. One, you get an allosteric inhibition of the PCSK9 binding to the LDL receptor, lowering LDL receptor degradation. This will increase LDL receptor expression in the liver and decrease LDL cholesterol in plasma. So that's really good. Another thing is you might directly inhibit PCSK9 binding to the LDL particle. If the molecule is binding to the ApoB binding site, it's also going to have this effect. This would decrease the ability of the furin-cleaved PCSK9 to bind to oxidized LDL, which again could lead to beneficial effects to decreased inflammation. We might also inhibit the linkage of ApoA to LDL particles. If this involves the direct binding of PCSK9 to the LDL particle, as we hypothesize. So this could also lead to decreased production of LPA. So we think the targeting of the LDL binding site in PCSK9 could be a novel way for both cholesterol lowering and lowering inflammation and atherosclerosis. Right. So with that, I'm going to end. Time 1 minute too long, and thank all these people who did the work, as well as my collaborators here at the Heart Institute and other places who especially helped me with the structural studies. It's funny.
Thanks, Tom. That was awesome. And I'm going to have the privilege of asking the first question. I was trying to just Google it quickly, but have the levels or activity of furin itself in inflammation been characterized?
Yeah, it is. Yeah, it is elevated as well. And so there were some ideas of inhibiting furin as well in treating cardiovascular disease and other related inflammatory disorders. Yeah,
but presumably, I mean, it's
harder to do though because it has so many targets, so there'd be pleiotropic effects of specifically targeting furin.
And that but that maybe is that's your hypothesis of why the cleavage is increased in inflammation is through increased activity.
Exactly. So it's sort of a feed forward thing,
right?
That the furin goes up, so there's more likelihood, and we see that in the LPS treated mice, perhaps, as well as the high fat diet treated LDL receptor knockout mice. You can see, that could be because of increased active furin as well.
And then just another quick question. I'm trying to even articulate my own thoughts. So pardon me as I think about this and ask questions at the same time. You're watching it all process live in my brain. But lipopolysaccharide is a lipid. Confirmed, right? Is that right? It is. Okay, good.
Bacterial lipid.
Just checking. I mean, I know, but I'm just, does PCSK9 bind LPS directly, or does it have? The reason I ask is because you hinted, like, is PCSK9 a DAMP? Like, if it's binding oxidized LDL on the furin cleave form, like, it's all suggesting that there's an evolutionary, as you mentioned, reason why we have PCSK9. And I think that's the biggest question everyone always asks: why do we even have this protein? It just seems bad. And so, what's the point? Well, obviously, there's a point. So, do you think that there's some, the biochemical interactions you were showing about the hydrophobic regions, et cetera, if you were to pivot that to say, instead of lipids in a lipoprotein environment, maybe lipids in a bacterial environment, is there anything?
Yeah, it's possible that it's we can do all kinds of studies like the liposome binding, like add oxidized phospholipid, add LPS, to see if that attracts the furin cleave PCSK9 to the liposome. Yeah, so those are really important things that can be done. LPS is transferred to lipoproteins.
Exactly.
Through transfer proteins, and then those lipoproteins are cleared by the liver. So that's an important role of lipoproteins in innate immunity. So clearly PCSK9 could be acting in this.
I'm wondering if
not only through lipoprotein clearance,
but sensing of some kind,
sensing thing, and then directly going to the inflammatory site. So,
exactly.
Yeah.
We have one question online before we get to you, and Ben. It's a good one. It's why are we systematically testing our patients for Lp(a)?
Well, that's because as I said, there's this clear connection of Lp(a) levels to cardiovascular disease risk.
Yeah.
And think about its properties. Like it has its prothrombotic activity, pro-inflammatory properties. It's sink for oxidized phospholipids. It's very enriched in cholesterol. So it's all around bad in terms of various mechanisms. And there's lots of genetic data associating elevated levels of Lp(a) with increased risk of cardiovascular disease. And now this, as I mentioned, this landmark study that came out in New England Journal of Medicine that it can predict a 30-year risk for cardiovascular disease. So that's really stunning.
Okay.
Tom, thank you so much for a great talk. I have two questions. First, so in a healthy person, in the liver, do we know on average how many times can the one LDL receptor cycle back to the cell membrane before it is degraded, mediated by the PCSK9?
Yeah, well, studies in Goldstein Brown lab years ago estimated that it could do that 100 times. So an LDL receptor could theoretically clear 100 LDL particles before it peters out. So PCSK9, destroying it, one event could again potentially prevent 100 LDL particles from being cleared in the liver. So that's why PCSK9 is so potent in raising LDL levels.
Okay, I'm asking this question because I'm thinking if PCSK9 has a physiological role, like for example, we don't allow very old cars to go back to the street because they are not safe, right?
Yeah.
So if probably one role in the liver is to a quality check, right? So to force the liver to produce new receptors.
That's an interesting idea. Like a lot of cell surface proteins, as they become damaged, tend to aggregate. And it's known, at least for the LDL receptor, that if it aggregates, that alone causes it to be degraded. Like if you could give a multivalent ligand or a VLDL particle, which has ApoE, which has many ApoE particles on it, so it causes LDL receptors to kind of come together. And that alone can cause the LDL receptor to be degraded. But yeah, PCSK9 could also have a role like that. I skipped over the slide, the physiological one, because I was running out of time, but I think it could have a role also in the fasting feeding cycle, where under conditions of low nutrition, you want your VLDL particles, your triglyceride-rich particles to circulate for a long time because they offload fatty acids to peripheral tissues. So they acquire apolipoprotein E during this process. So the LDL receptor in the liver can contribute to the early clearance of those triglyceride-rich particles, which might not be a good thing you want under conditions in which you're undergoing prolonged fasting, for instance. So an evolutionary role of PCSK9 could be in sort of maintaining these circulating lipoproteins, these triglyceride-rich circulating lipoproteins. But that particular role we definitely don't need anymore with a high-fat Western diet. We don't need anything to protect our circulating lipoprotein levels now. Yeah.
Okay. Do I have time for another question? Okay. Thank you. So the classical role of the PCSK9 is in the liver. So it is secreted by the liver cells and act locally. So if we are studying the PCSK9 in the bloodstream, there may be a gradient at different vasculatures, meaning like the blood leaving the liver would have a highest level of PCSK9. Actually, I don't know, just some thoughts. Is there any correlation with the risk of the atherosclerosis based on the predicted gradient?
Well, yeah, so there's two contributors probably to the atherosclerotic risk. And one is the LDL, one is the liver-derived PCSK9, but it's also produced, they show in endothelial cells, macrophages, I think even produce it, but endothelial cells for sure produce PCSK9. It's upregulated under inflammatory conditions in cultured endothelial cells. So there's this local concentration of PCSK9 as well, which could even be higher than, we don't really know, right? We don't really know what that local concentration is. It could be even higher than in blood circulation. And they found it in atherosclerotic plaques. They find PCSK9. And there was one study in mice, even in where they knocked it out in the liver specifically, they still saw PCSK9 in the plaques in mice. So there's also locally produced PCSK9 under these pro-inflammatory conditions. So it seems to have these dual roles, one in regulating LDL cholesterol and the other in regulating inflammation. And given the interconnections of the two, it kind of seems like it's a natural sort of player in both, that it can regulate both the cholesterol and the lipoprotein aspects of innate immunity, as well as having maybe some direct effects on the immune cells.
Thank you so much.
And there's one question online about whether LPS affects LDLR recycling and whether or not LPS affects PCSK9 binding to LDLR.
Yeah, I don't know. I don't know that that's been looked at, but definitely LDL receptor is, I believe, downregulated during inflammation. And one of the things that happens in inflammation that's interesting is reverse cholesterol transport is inhibited, and one of the effects of that is to raise cholesterol levels in the immune cells. And because cholesterol in the plasma membrane is very important for signaling platforms known as caveoli. And so cholesterol buildup kind of helps those signaling mechanisms along. Exactly, so the reverse cholesterol transport that we studied is downregulated. And one of the ways is again, like LDL receptor in liver is downregulated because that prevents the sort of sucking of the cholesterol out of the peripheral tissues back to the liver. Yeah.
Awesome. Okay. Thank you so much, Tom, and thank you, everybody.